All right, and welcome back to Calling Out Controversy here on WVCR 88.3 Saint. I'm Justin Kenyon, and with me tonight is Ben Novak from Revive and Restore. So, Ben, thank you for being with us.
Yeah, it's great to be here. Thank you for having me.
So, why don't you start us off by telling us a little bit about yourself, Ben?
Yeah, so I am the lead scientist for Revive and Restore, which is a conservation nonprofit with a mission to advance the innovation and adoption of biotechnologies in conservation. Our shorthand way of saying that is we are bringing genetic rescue to endangered and extinct species. Our more well-known and controversial projects are our long moonshots to recreate extinct species like the woolly mammoth and the passenger pigeon, put those back into the environment. Um, but in the last few years, we've been able to really grow as part of all of our staff's work into a much broader organization thanks to our new funding sources and our ability to actually provide funding as a partner. And a lot of the work that we do now in 2021 is, as I said, fostering the innovation and adoption. We're able to actually really fund and spearhead global projects in conservation genomics. And we're working more with endangered species than we are extinct species now.
So, I guess the question that comes from all that, especially for myself and people who are not really familiar with your mission, is why? Why try to bring back endangered or extinct species from the dead?
Yeah. So I mean, the biotechnology, it can play a really interesting niche in conservation. And a lot of people, when they hear de-extinction or biotech for conservation, they think of it, in this completely new and alien thing. And it's very important to see it in the context of what conservation has been doing for over a century. And so when it comes to, say, restoring um, lost species in an environment, that's actually something people have been doing since the early 1800s for various reasons. One of the very first projects to restore an extinct population was the capercaillie grouse in Britain. In the 1830s, at the early 1800s, capercaillie were extinct in Britain. And in the 1830s, a team of people decided to bring capercaillie from Europe to the UK, to Britain to restore the population. And that was mainly done for hunting purposes. But over the decades, that practice of restoring locally extinct populations became more and more utilized and more driven for restoring just natural ecosystems, culminating in probably the most famous example, which is the return of wolves in Yellowstone in 1995, which has had huge benefits to that ecosystem. And so, our goal, our vision is that people have been doing this a long time for many purposes, everything from returning ecological dynamics to bringing in new economic opportunities in systems. But when it comes to species like a mammoth or a passenger pigeon, there's no living relative to draw on to put those animals back in their habitat. So it needs some new technology to be able to do that. And many technologies came together to do that. But every technology that there is to recreate something like a mammoth or a passenger pigeon, every step along the way is a technology that could be immediately used to help the conservation of an endangered species, which are facing threats that are more and more intractable to conventional conservation measures. So it's really about bringing in and augmenting tried and true conservation to overcome problems that are becoming worse and worse.
So, I mean, when I think of all this and I think about bringing back endangered species or, for other cases, extinct species, and you reintroduce them into ecosystems, like you brought up the 1995, was it where they reintroduced wolves into Yellowstone, correct?
Yep, yep.
Now, are there any cases where like they've been so far removed from kind of that ecosystem for decades or hundreds of years or what have you that they kind of act instead of like rebalancing the ecosystem, they kind of act as an invasive species because they've been away from that ecosystem for so long?
Well, I actually started doing research on that a couple years ago. because it's a question that comes up. It's a good question. There are many cases in which, of course, when you introduce a new species to an ecosystem, it becomes invasive. At least about one out of every, say, 100 or so introduced species will become problematic. So yeah, the question begs, like, how long can a species be gone before it's brand new to the ecosystem? And it's a complex question, but what we've found in our research is that so far, of all the examples we've seen that are driven for conservation, there's been no case of a reintroduced species becoming problematic in its ecosystem. There have been some issues where a reintroduced species might start to overpopulate, but that might be because there are no predators for it anymore. So you either have to reintroduce the predator or bring in some managed hunting on those situations. But in general, over the world, reintroductions of species that have been gone for several hundred years to sometimes several thousand years have not ended up badly. They've actually done really good. But it all comes down to careful planning and looking at what the ecosystem is today and what will stand to benefit by bringing in species that once lived there in the past. It almost never ends up creating some historic version of an environment. It's always a new modern environment, but we look to those past species to get an idea of what worked here before and can this work again. And it turns out that it's working pretty well. The longest one I know of was in 2017. European bison were reintroduced to Spain, and they haven't been there in 15,000 years.
Oh wow, that's definitely
The
long time.
the wolves in Yellowstone were only gone for 70 years.
Yeah, not that big of a difference when you look at it in the grand scheme of things of environmental history.
Yeah.
So I mean, but then again, like you think about it, where it's just like a woolly mammoth has been away from its ecosystem for hundreds of thousands of years, which would be different than bringing back a passenger pigeon that went extinct in, I believe, 1914.
Yeah, well, maybe that's the thing is so mammoths went extinct virtually everywhere in their range between 10 and 7,000 years ago. They did live in some islands until 3,700 years ago. So they were alive in historic times, but just in these tiny little isolated islands where they were surviving. And that's a big, that's an order of magnitude difference to the 100 and, now 7 years that passenger pigeons have been gone. But we can think of this in terms of more recently extinct species. So there are a lot of species that have gone extinct in just the past few decades because their habitats have been degraded or due to, pollution problems. Things like the Yanks River dolphin went extinct probably by 2010. And you could not return Yanks River dolphins, even though they just went extinct less than 20 years ago. You could not put those back into the environment because the environment is still not recovered or safe for them to be there. The pollution and fishing problems that drove their extinction are still being done in the Yanks River. Now, the passenger pigeon, different story. It's been gone over 100 years. And in that 100 years, its habitat has actually been growing back. So forests in the eastern US hit their lowest point between the 1920s and 1930s. And owing greatly to agriculture moving out to the Great Plains and mineral resources being tapped out and energy and resource economics changing, lots of land that was deforested in the eastern United States was left to become forest again. And there are areas in the eastern US where they have as much forest cover today as they did in the 1850s, and some places have as much forest today as they did in the 1600s, particularly New England. So that's actually evidence where the longer time has been able to pass, the habitat has actually been able to come back and be restored. It's now at a point, though, however, that there's a lot of forest cover, but there aren't the right dynamics happening in forest regeneration and turnover to actually maintain and keep most species that are there. So that's actually the biggest reason why we now, after this century of regrowth, we want to bring passenger pigeons back to kind of complete the picture, so that all those species that live there continue to live and thrive the way they did for hundreds of thousands of years before. Now we go over to the mammoth, it's a kind of a different story. The extinction of the mammoth actually, and bison and deer and other grazers in the north actually brought about the extinction of their habitat, the grasslands, the mammoth steppe from there. So technically there's no environment to bring a mammoth back to, but because mammoths and grazers were actually crucial to engineering their environment, there's research going on at a place in Siberia that's been going on for decades, showing that you can return grazing animals to the tundra. And if you supplement their feed for about a year or two, they will graze upon what limited grasses are in the tundra, and their activity will start to favor grasses to other plants, and they will actually engineer and create the grassland ecosystem. So that grasslands can be restored, but you actually have to bring in animals like the mammoth to do it. So those time frames, it doesn't really matter how long a species has been extinct, as much as what has been happening in the environment in that time frame and what can happen by bringing in the species. So it's always about the dynamics.
Okay. We're here with Ben Novak from Revive and Restore here on Calling Out Controversy 88.3 Saint WVCR. And so with extinct animals like woolly mammoths, how exactly, especially since they've been extinct, with the most recent being 3,700 years ago, how have you been able to get a hold of anything that would lead to them being brought back, whether it be like cell walls or anything else? Because I know there are some cases where like they've been pretty much just like frozen with all literally everything from their muscle to their bones, but then is that even usable?
That's a great question. So yeah, I mean, every few weeks it seems there's a news post of some frozen, cave lion or Pleistocene wolf or mammoth, leg or something that comes up out of the permafrost. And yeah, it's got muscle and hair, and it's frozen. And those things have been frozen for thousands of years, but sadly, just because they were frozen, they were still deteriorating at a microscopic level. So the molecules inside the cells are still breaking down, and their DNA is highly fragmented. Just like what we get for passenger pigeons, we take their museum skins, taxidermy skin, and you can use that just like a frozen piece of mammoth. You dissolve it down to get the DNA, but in both cases, whether it's been frozen or sitting in a drawer, the DNA is heavily fragmented. This is both a pro and a con. So the fragmentation actually makes it easier to sequence the DNA. We can sequence lots of mammoth DNA or passenger pigeon DNA, or really any extinct animal we can get DNA from, as long as the remains have been kept in some kind of condition, good condition, either cool, frozen for thousands of years. The oldest DNA that anyone has gotten yet is just over 1 million years old from a different species of mammoth from the Pleistocene. But we can get DNA from just about any taxidermy piece you see from a museum, but it will be heavily fragmented. From those fragments, we can piece back together what the code was by comparing it to a living relative's genome. But the thing is, since it's fragmented, there's no hope of actually taking cells from a frozen piece of mammoth tissue and cloning a mammoth. To do something like clone an animal, you need living, intact cells. But what we can do, which is what George Church's team has started doing with with the Mammoth Project, is we can take the cells of a living relative, the same one that we use to compare the DNA to, and we can start using something called gene editing to rewrite the DNA code of a living animal to have the DNA of the extinct animal. And we can't do that at the scale of the entire DNA code, but we can target genes, dozens to hundreds of genes that are involved in the key ecological traits that are going to allow us to restore an animal like a mammoth to the tundra. So in this case, Asian elephants are already almost identical to woolly mammoths. They're the same size. There's even scientists that can get that when looking at old elephant bones will get confused on which is mammoth and which is elephant. They're so similar. But of course, a living Asian elephant cannot survive the winters of northern Siberia. So it needs to get the same adaptations that allowed mammoths to do so. So that's the goal of the project, is to use mammoth DNA to figure out which genes were involved in making the wool, the layers of insulating fat, which ones were involved in working with blood that binds oxygen at lower temperatures better, and cells that work better in lower temperatures, those things, and giving those traits to an Asian elephant, so that basically we're making a woolly Asian elephant that is now capable of surviving in Siberia, and that is the animal that will replace the extinct woolly mammoth. And for passenger pigeons, we'll use, in another similar case, we have bandtail pigeons, which are extremely similar. They eat the same things. They're about the same size. They're basically the West Coast version of a passenger pigeon, but they differ from passenger pigeons in just certain traits that are important for what the passenger pigeon was doing in the environment. So we need to be able to use this gene editing and the passenger pigeon's DNA code to be able to make a living hybrid, basically, that has the same ecological role. But when we do have intact living cells that have been preserved for years, we can actually, avoid all of that complicated gene editing work, all of that stuff, and potentially bring something back directly. And that's something Revive and Restore just recently did with black-footed ferrets. So black-footed ferrets are an endangered species. They are not extinct, but they almost went extinct. In the mid-1980s, there were so few of them left in one place in the world that conservationists went into the wild, gathered up as many as they could to bring them into captivity to save them, and have since 1987, they have bred nearly 10,000 black-footed ferrets. They've reintroduced them to 30 locations throughout their old range. There's a long ways to go before this species is fully recovered, but the 20 generations and 10,000 ferrets they've bred are descended from just 7 individuals. They have a tiny gene pool. And today, every black-footed ferret is basically a sibling or a first cousin. That's how closely related they are. But back in the 80s, when they were capturing ferrets, a few very forward-thinking people took some tissue samples and sent them to San Diego Zoo, where Oliver Ryder took those tissues, made cell cultures, and froze them away. And it turns out that those were a couple of ferrets that are unrelated to all living ferrets. They're not among those 7. They died before they could produce offspring in captivity, and they now are, because they preserved living cells, intact cryopreserved cells, we can use technology like cloning to bring those individuals back from the past into the present day population, to bring in some new genetics to help out that small gene pool, which is exactly what we've done with the news that was released back in February, that we cloned Willa, the ferret, from her cells that were preserved in 1988. And Willa, her clone named Elizabeth Ann, is now going to be an 8th new individual into this gene pool of 7. And she's doing great. She's over 110 days old now, and she's in perfect health. And that's not the first time we did that either. We actually cloned an endangered horse, the Przewalski's horse, which has the exact same history. It went extinct in the wild. Just a few animals were left, and now they've been recovering them. We cloned an individual whose cells were preserved in 1980, so even longer ago, he's not a brand new individual in the population, but he's so many generations separated, and that we're still bringing in some diversity that's been lost. So, I mean, that goes back to what I was talking about earlier, is, every step along the way for this type of de-extinction work is something that you can harness and bring in for an endangered species right now to help some problem that they have. And if you have living cells, it makes it a lot easier.
We're here at Ben Novak from Revive and Restore here on Calling Out Controversy, WBCR 88.3 The Saint. You actually provided me with the segue that I now no longer have to do because you actually provided it for me, which was I was going to start asking you about Elizabeth Ann. And Elizabeth Ann, for those who kind of loosely follow this segment of whether it be genetics or biology or conservation, has probably been the biggest news of cloning since Dolly the Lamb back in the 80s.
Yeah, I think that's arguably correct. We actually, the San Diego Zoo did some analytics of the press that's come out on Elizabeth Ann, and the news stories from the New York Times story and the many stories that picked it up have apparently been seen by 1.36 billion people in the world. And it was amazing to see the response. It was overwhelmingly positive. Dolly the sheep is the world's most famous clone in history, and her story is mixed between people celebrating her and people that came under a lot of misconceptions about how cloning works based on her story. And to see nearly 25 years later, this kind of first big event for conservation gain notoriety and be well received was it was really overwhelming to all of the partners involved in that, from U.S. Fish and Wildlife Service to San Diego Zoo, Viagen Pets, and of course Revive and Restore and the recovery team for the black-footed ferrets. But it was a long project in the making. We started that effort back in 2013, and it was proposals and paper and discussion for years before it ever became real science, including sequencing the genome of Willa, the ferret that we were going to clone, to make sure that she was actually going to bring in new genetic diversity to this population. And it turns out, yes, when we looked at her genome compared to living ferrets that are not only 33 years removed from her, but also unrelated, her genome has as much as 3 times more unique variation than any living black-footed ferret. So today, her clone, Elizabeth Ann, is the most genetically diverse and valuable black-footed ferret alive. And we're all extremely excited to see how this work continues to help that species, because there's a lot more. This is just building block number 1. We have a much longer genetic rescue program with a lot of research going on to help black-footed ferrets overcome not only their genetic diversity problem, but the other threats that face them in the wild.
And I must ask, where does the name Elizabeth Ann come from?
It was just on the list of eligible names. There's no story behind it. I mentioned earlier that, of course, since 1987, the recovery team has bred over 10,000 ferrets, and I should say they'll hit 10,000 ferrets this breeding season, which is starting right now. And that's a lot of ferrets, and every single one has a stud book number, but along with a number, they also have given every single one of them a name. So at a certain point, they've just started keeping a list of names that haven't been used yet, and Elizabeth Ann wasn't taken.
Oh yeah, because here I was thinking there's like some meaning behind the name that they just kind of gave it, and I guess the same could be said for Dolly the Lamb too.
Yeah, I mean, there was nothing truly significant. Sometimes people will give a clone something, a fun name, like the world's first cloned cat was named Copycat. So that was nice and clever, but usually
Nice
it's
pun.
usually after somebody who was involved in the team or in commemoration. The clone of Przewalski's horse was named Kurt after Kurt Benerschke, who was the founder of the San Diego Frozen Zoo, and who was actually involved with saving the cells from Kurt's original donor, whose name was Kuporovich. And so Kurt has a special story to his name. Elizabeth Ann is just a name that hadn't been taken.
Now, another question I must ask, because I know there will be some people who kind of think this is just some wacky mad scientist type thing where it's kind of just like one of the big things that comes up in bioethics with some people is the notion of playing God, and that this is something where you shouldn't do it because you're kind of messing with nature. And when you hear comments like that about your work or the work that other people in your field do, what's kind of your response to that?
Well, you know, there's several responses. The first I'll offer, of course, is that biotechnology is in no way playing God. It's actually human beings trying to get better at um, at working with nature. Everything we use in biotechnology, we take from something we've discovered in nature. So for those that believe that nature and the world is God's creation, we are not at the level of being able to create the way God did. We are actually using the tools that God created to try and help stop destroying his creation. So like CRISPR itself, gene editing itself, comes from bacteria. Um, and the track record of genetic engineering and biotechnologies is actually very good, in both in agriculture, when it comes to in terms of the safety of the organisms and what's going on with that. Now, when it comes to the social dynamics of how these things have played out, it's very different. It's created, that's where a lot of the controversy has come from. But for the organisms themselves, we are, the motivations are and the outcomes have been very different from what a lot of people think. And that's where the second answer to the question really comes in, is that conservation has been working to undo unintentional as well as ignorant and intentional damage to nature for a long time. And there are lots of wins. We too often confuse things that have damaged nature with things that are intended to help nature. So, a common case is people will point out, oh, we shouldn't mess with nature because, well, look at the cane toads in Australia. The cane toads were introduced in the 30s and they've wreaked havoc ever since, and they're overpopulating and causing issues. But when someone actually looks at that story, cane toads were introduced as a biological control to try and control a pest to sugarcane crops. They were not introduced to help the environment. They were introduced actually by people that didn't even regard the environment they were doing it. And actually, even though they were introduced by a government agricultural agency, Australia's scientists at the time warned the government not to do it. So hidden in these horror stories are a lot of context that actually shows that they are examples of why we now have to do good work rather than examples of meddling in nature becoming bad. And actually fast forward to today, and today biotechnologies are now the first solution that offer a potential means to bring the cane toads back into check. There are actual friends and colleagues of mine at Australia working on a genetic engineering solution to be able to control cane toads. And these types of interventions to then go in and help a problem have been built on years and years and years and decades of people going in and fixing problems in nature and doing really well at it. So if the answer is twofold, either one, if you do subscribe to the belief that God created the world, then we are in no way as powerful as God, and we are not playing God. We are using God's nature he created to try and preserve the nature he created at it. And as long as there are not complexities that get in the way, things that are often driven by political dynamics or problems with the human side of things, we can have really good results. And we have been saving species for a very long time using interventions of every kind, from captive breeding to reintroductions and translocations to mixing populations to help their genetics to using biocontrols actually for good and for conservation. There have been many examples now since the 60s where biological controls have been used to actually control an invasive species and help save endangered species and environments.
Okay, and then coming off that, now that we've discussed also a lot of species that have been cloned and or in the works of being cloned, what do you think is kind of the most interesting species, whether it be endangered or extinct, besides the ones that we've talked about being like passenger pigeons, woolly mammoths, black-footed ferrets, that people like you hope to clone and bring back, or are we in the works of cloning?
Oh, well, that's a good question. No one's asked that one. I mean, I usually get asked, if we're going to do de-extinction, what would be some interesting animals to do? But of course, that's the idea of using the gene editing and
Yeah.
the full de-extinction tool set. And there are a number of extinct species, I think, that would be, number one, just beautiful and amazing to have back in the world, but others that would have valuable roles in their ecosystems today. There are people interested in bringing back the Tasmanian tiger, more appropriately known as the thylacine, and there are people interested in bringing back great auks to the North Pacific. People interested in doing quite well, but there's the argument to be made that gene editing could actually resurrect those traits much better. When it comes to cloning for conservation, like what we've done with black-footed ferrets and the Przewalski's horse now, I think there are a number of exciting prospects. Probably one that's pretty far out there that I think would be really good would be to clone the critically endangered vaquita, which is the world's smallest type of whale or dolphin. It's a porpoise that lives in the Gulf of Mexico, and it is endangered for many, many complex reasons. But the main thing causing it to decline is fishing going on in the Gulf. These porpoises get caught in the fishing nets and die. And of course, the fishing is crucial to sustaining the local community. So it's a difficult situation. For years, people have been trying to capture vaquitas to establish a captive breeding program to prevent them from going extinct. But vaquitas become so stressed when they're captured that they die, or they have to release them, before they die. Because they just can't handle being handled by people. And so there's a huge argument there where if someone could get their hands on some vaquita tissues, we could clone vaquitas into captivity. And the reason that would be a huge leg up for them is we would use something like a bottlenose dolphin to be the surrogate mother. And an animal born into captivity is not going to experience the same levels of stress in captivity and being handled by humans that a wild caught animal experiences. And this is true across the board, even though other species are much easier to capture. And the nice thing is that captive born animals have shown not to have the problems going back to the wild that wild animals experience going into captivity. So you could actually clone a population of vaquitas in an aquarium, basically, to keep that species from going extinct, and then use the offspring of clones to start restocking the wild population. And I think that would be a really amazing use of the technology. I think there's actually a lot of uses in which cloning animals from the wild to bring them into captivity would be an optimal use in the future, so that we can leave animals in the wild so that they can continue to breed and hopefully possibly recover in the wild on their own. But we're creating backup populations in captivity in case we lose those individuals in the wild.
Okay. So I got one final question for you before we end this interview. And that is, what is your favorite thing, or favorite little factoid or tidbit or story that people may not know about your line of work that you really enjoy? Or it's kind of something that you kind of like to talk about that most people don't really know about your line of work?
Oh man, you've got the best questions. I have to say it. I don't get asked these very often. I mean, it's hard to pinpoint one because I'll bring up lots of them. There's tons of examples of my favorite are to bring up examples of conservation success. because they don't get talked about as much as the problems facing conservation, and there's a lot of them. There's a lot of problems, but there's a lot of success stories. So, one interesting thing, of course, is that wild turkey were extinct in New England for 160 years, longer than the passenger pigeon has been gone, and in the 60s, 70s, 80s. And so there are populations, like the population of turkey in Connecticut and Massachusetts is younger than most people living in Massachusetts and Connecticut. But today they're so ubiquitous that people don't even really remember that history, that they were gone. There's a lot of cool stories out there. Even stories of when, I think this is a neat one, an unintended consequence actually becomes a benefit, right? So people often think of meddling with nature as always having these damaging unintended consequences, when in fact sometimes something that people didn't mean to happen ends up being a good thing. So in Canada and Montana, a hawk moth, the leafy spurge hawk moth, was introduced as a biological control for leafy spurge, the weed. And this was for agriculture. It was not a conservation intervention, but the leafy spurge hawk moth spread from Montana into North Dakota, which is actually my home state. And by the time it reached eastern North Dakota, it hit some nature preserves, which were the home of the last populations of the endangered western prairie fringed orchid in the state. And this orchid has a highly evolved relationship with North American hawk moths. And amazingly, even though the leafy spurge hawk moth is from Asia, it has the right kind of mouth parts and diet that it actually started pollinating the endangered orchid, and is today the #1 pollinator of a native endangered flower. So this introduced animal from Asia, the population of a native endangered flower. And there's a lot of cool, cool stories like that out there. But of course, for anybody that wants to really delve into this, I've actually just published a paper on translocations. It's called U.S. Conservation Translocations, Over a Century of Intended Consequences. It's open source. It's free to find online. It was published in Conservation Science and Practice. I've also written a paper on what is de-extinction, which is also open source. If you just look up Ben J. Novak de-extinction, it's in the journal Genes. And of course, Revive and Restore's website is a great place to go to find out more about the projects we work on. And I think every project is a cool, basically it has a cool tagline that I'd love to share. So definitely check that out. And of course, we are a nonprofit. So all of our projects, whether or not it's funding, the genomics to help breed disease-resistant sunflower sea stars, or to fund the first conservation genomics project ever in the country of Bolivia, or if it's returning Xerces blue butterflies to San Francisco, all the way to the de-extinction of the woolly mammoth, like this, and as well as cloning historic black-footed ferrets like Elizabeth Ann. All of this is made possible by the donations of everyday people to our organization. And a donation of $10 or $20 or $100 might not seem like much, but every single small donation like that is what ends up bringing some beautiful genetic rescue effort to fruition. So if you're interested in supporting the work, to any of the viewers, go to our website and please donate, and know that your money will go directly into something.
All right, and this has been Ben Novak and Justin Kenny on Calling Out Controversy, WVCR 88.3 Saint. Ben, it's been a pleasure having you on. Thank you for being with us.
Thank you.